Okay, my name is Choong-Wan Woo. I go by Wani. We have only 20 minutes and two talks, so I'll be a little bit in rush. Sorry, I don't have time. Okay. So, I put this slide. Sorry. This is a quote from Dr. Peter Bendettini's blog many years ago, but still echoes in my mind. We are just one clinical application away to see another surge in fMRI studies. I really want to make this happen, and I think personalized functional mapping has a great potential. And this quote is also from this paper, recent review paper already Tim showed us, a really nice review. This paper, the authors lay out these three main areas that PFM can contribute. But I think I'm going to be really quick on showing linking brain functions to, we are seeing great papers, great studies on this area, and then we are seeing many papers. And if you haven't checked this preprint, awesome preprint on personalizing DBS stimulation targets for pain, is a couple days ago, I think it came out. But for the last one, tracking clinical and treatment progression, there are not many papers. And we are working on this area. And I want to convince you that this area is also really important. This table shows the multiple types of biomarkers. And among them are actually targeting the within individual changes. And this is particularly important for drug discovery because you know the strong placebo response get in the way of developing new drugs, and partly because we don't have any alternative measures other than self-report-based outcome measures. So we are really working hard to make surrogate markers based on the brain for pain. So that's why we launched this MPC project. It stands for My Pain Connectome Project, or Midnight Pain Club project. That doesn't mean that we gave pain to participants in dark midnight, but we named it because we were inspired by these two projects and My Connectome Project and also Midnight Skin Club project. But our aim was different. We aim to develop examine the upper ceiling of the accuracy of fMRI-based prediction of pain. So it consists of two different data sets. One was MPC-1 that involves extensive sampling over one participant, multiple scans, and the other one was MPC-100, cross-sectional study, many participants, but single scan. N equals 1 participant was me, so we call it MPC-1 as well. And among them, 28. It had 33 sessions. Each session lasted 2 hours, so almost like more than 60 hours of data. It had 28 awake session and 5 sleeping session. I don't have time to explain the sleeping session, but it's interesting, right? Getting pain while sleeping. Okay. But we had two types of pain, heat pain and oral capsaicin. I'm going to focus on the heat pain today. We had 124 participants with the same design for MPC-100. We aimed to predict single trial level variations in pain ratings. And so with the single trial beta maps, we divided the data into 3 parts: training set, validation set, and testing set. We used the training set around 60% of data to identify the pain predictive patterns, and then we used the validation set to fine-tune the hyperparameters, and then we freeze the weights, all the predictive weights of predictive maps, and then tested those models on the independent test set. And we had 3 different modeling inputs to predict one person's pain, trial level pain ratings. One was population level prediction. So using the other people's brain pain model and test that on my own pain ratings. And then the second one was personalized brain model. That is using my own pain model and then predict my own pain rating. And the third one was a little bit unique in this analysis. We used experimental parameters, including stimulus intensity, skin sites, and trial orders. So you would expect that we can achieve really high accuracy if we use these experimental parameters, because pain ratings is largely driven by this heat stimulation, right? And all the results that I'm going to show you is based on this testing on this independent testing data set. The results can be summarized with this Venn diagram. Using these three modeling inputs, we were able to explain around 68% of variance of single trial level pain rating. You might not be surprised, but I mean, I was really surprised by this number, because I've been seeing many numbers for the last 15 years in predicting pain rating, but this is really highest number that I saw. And R-squared only with the brain was 61% or so. And if we decompose the variance explained by population level model versus personalized brain model, you can see personalized brain model actually explain most of the variance, and then population level model, even though that explained around 26%, it doesn't have an additional kind of value in predicting trial by trial rating. And interestingly, when we use the experimental parameters, the R-square was around 0.53. And then that was smaller than personalized brain model explained variance. And there was a unique variance explained by personalized model, that almost like 12% explained variance. And so this interesting observation that personalized model can provide prediction above and beyond the experimental parameters really made me think that a brain-based biomarker is capturing the internal factors, variances induced by internal factors like maybe endocrine system or other autonomic systems, in addition to external factors. So I think it's an important kind of point. And how much data do we need? We need if we use the whole data, this is the accuracy. This is one session accuracy. But maybe we might be able to find the sweet spot that could be maybe around 5 sessions or so, 50% explained variance. And if we see compare the personalized model versus population level model, actually it shows kind of similar regions, but if we look at the pattern similarity, actually they were not that similar. The correlation was really low. And then that means that maybe there could be fine-grained pattern differences, and then when we apply the smoothing kind of kernels, the effects, deteriorating effects, was larger for personalized model. That means that the personalized model relies on more fine-grained pattern information. And also, as you kind of showed, as Tim showed earlier, scan network inter-effector regions is known to be, correlated with pain, right? And then, interestingly, personalized models show that inter-effector region really clearly, but not in the population level model. So, personalized models show higher sensitivity in identifying pain predictive brain regions. And if we look at the important regions, actually higher order brain regions were important for personalized model. Okay, and we had a 5-year follow-up. So I went into the scanner again this year, and then you will see the details in this poster. And yeah, I don't have time to explain. And we also had, in the meantime, we aimed to develop personalized marker for patients with chronic pain. So we scanned actually 3 patients with fibromyalgia, but only 2 out of 3 completed more than 15 sessions. So we analyzed those data. We had many tasks, but I'm going to show you briefly about the prediction performance for the spontaneous pain rating. We had 3 runs, and in between run 1 and 2, we had an individualized pain exacerbating physical maneuver. So pain increased and then decreased. So over time, we had this continuous pain rating and developed the marker based on connectivity. So kind of long story short, it worked. Worked. And then we need more than 5 sessions. And cross testing didn't work. Okay, so individualized model important. Okay, it's 10 minutes. Okay, I'm doing good. This is last slide. So we are working. Our lab is working in this area, but I really need. I came to think that I need to make a connection with these other areas, like to understand the neural mechanisms underlying these pain features in chronic pain patients using this PFM method, and also personalize the treatment targets. So we really want to make kind of one clinical application early. Thank you. 10 minutes.
Since Wani did such a great job trying to stay on time, maybe we have time for just one brief question.
I mean, I prepared the 15-minute talk.
Okay.
Okay. If you have a question.